My name is Julie Klaus. I'm the CEO and co-founder of Aptabridge Therapeutics. And I'm really excited right now to share our mission to build the next generation of cancer immunotherapy. As you know, cancer is a global challenge that affects millions of people every year. Lymphoma is the 5th cancer killer globally, and an estimated of 550,000 people will be diagnosed this year. That's over 1,000 people per day. Even more concerning is that these rates are going up, with developing countries expected to see as much as 80% of increase in the next decades. And if you look at the US alone, you have 20,000 patients that will die because they lack effective lymphoma treatment. Many of us know how devastating a cancer diagnosis can be for patients and the people around them. I've seen this firsthand in my family, and it's frustrating to see that our frontline therapies come with such huge side effects that are often not completely cured in the long run. To illustrate a little bit what we do, then, as you can see, you can use the analogy of the body as a map, where your natural immune cells, your white blood cells, can patrol the body via the roads and bridges. When cancer begins to grow, normally this immune cell will find the cancer and identify it as a threat and get rid of it. Your body is extremely good at this, but over time, cancer can evolve. And they can escape detection by breaking down the bridges. And this is called immune evasion. And to reactivate your natural immune system against cancer, we need a way to reinforce the bridge. And it's exactly what we do. We build DNA nanobridges to help your immune system find and eliminate the cancer. If I illustrate a little bit what we do, then you can see in the center bridging a human cell and a cancer cell, and each end recognizes a unique feature on the cell surface and are connected using DNA bridge that links ends to the immune cells and can eliminate the cancer cell. And we call that precision medicine. And you see better on this specific graph how we build the bridges between the two cells. To construct these therapies, we use a combination of molecular evolution and artificial intelligence. Molecular evolution is a laboratory process we use to identify the specific bridge ends. We can use this process to make specific ends, not just for lymphoma, but also any type of cell. That means we can substitute the cancer binding arm to target other cancer types. And we then use a proprietary machine learning program that Dr. Taylor Cottle developed to connect multiple binders. And we are currently drafting patents for 2 novel aspects of this process with our legal team at MINS. Here, just an illustration, when we mix the cancer cell binder with the cancer cells in the culture, we can see on the left bar, we can see this binder that binds the correct cell type at higher concentration. Now, why should we use this type of molecule? There is some important advantages using DNA and RNA. Compared to treatment on the market, DNA and RNA are easier to engineer and manufacture, which brings down the cost. And it's also less toxic and using well-studied biology. And you can see in particular using DNA, like dry DNA samples, and you can ship it everywhere, like in a paper envelope. And this is huge if we think about global access. Right now, we are raising $500K for now, as we are doing our proof of concept, but we are planning to raise 10 million by 2026 to enter in clinical trials. And we are currently in R&D phase, developing a new binders for lymphoma. And by 2024, our goal is to advance our best molecule to FDA-enabling experiments, which give us the data to request a human clinical trials from the FDA. And this is a critical point. If we can generate positive data, this is where a pharmaceutical company may want to either partner or acquire the company. We know this is a viable strategy because similar immunotherapy platforms to Aptabridge are generating huge value. IPO's value after partnership announcement are ranging between 400 and 500 millions with outright acquisitions over 2 billions. And if you look at the last JP Morgan Life Science report, they show that oncology is still the #1 driver of M&A and still a top investment for big pharma through external innovation. And actually, we have already been in contact with 2 potential partners that are interested to know a little bit more about our platform. The plan is to start with B-cell lymphoma, with a market of 6 billion in the US and 10 billion globally speaking. But it's not where we want to stop. If we have a success with B-cell lymphoma, it opens door to build Aptabridge therapies for lung and breast cancer, which represent 40 billion markets, and greater immuno-oncology target market, which represents at least 90 billion, with new targets still being discovered. But nothing would be possible without this team. Taylor and I met 2 years ago when I joined his team to participate in the accelerator nucleate between MIT and Harvard. We won the ECL award, representing around 80K to run our first in vitro experiments. And what I found amazing with Taylor is his ability to mix an amazing background in molecular biology. He just finished his PhD at Johns Hopkins and have over 10 years of research experience from cancer biology to DNA. And I've been able to convince Sebastian to join us. He brings more 10 years of pharma operational experience, starting a new drug and moving it successfully in clinics, as well as an incredible resilience. And to finish by me, then I bring more than 15 years growing in big pharma as an executive. I successfully advanced my career by having a high focus and shared vision, making sure we are all aligned on making the right decision without compromising the vision. And last but not least, we are also joined by an impressive board of experienced advisors building successful biotech, high-tech operational partners. And in all, I hope you'll help us at Aptabridge as we build accessible therapies to cure cancer around the globe. Thank you so much.
Julie, what a fantastic presentation. And congratulations on all your progress. It's fabulous. I have several questions I wrote down, but I'll open it up because, oh my gosh, you're doing God's work over there. So I'll ask everybody else if they have questions, and then I have like 15 questions, but are there any questions over there? Okay, so we're running a little bit behind, but may I please ask a question about your advisory board, since right there, I mean, regulatory, yeah, lots of who's your the Arthur Hiller person? I mean, there's too many questions wrote down. How do you feel about your business advisory board? Let's put it that way.
Yeah, then we have some scientists and experts in DNA nanoparticle like Rob Trapp, where we have regular discussion with. We have also specific experts that are more on the regulatory paths, because the plan is to go as smooth as possible with this new modality to a clinical phase. And it's super important because it's a new modality, and we want to partner with FDA and EMA as soon as possible with that. And we have also serial entrepreneurs, and Arthur is a CEO of biotech company as well, and is bringing also some insight on how to strategize our company and make sure we can deliver as soon as possible the first assets.